Hi, everyone, and welcome to episode 353 of your Tick Bootcamp podcast. The title of today's interview is The Lyme Rebel, an interview with Dr. Eva Sapi. My name is Richard Johannesson.
And I'm Matt Sabatello.
Folks, this is actually going to be a longer preview than we've ever given before because this was one of the most densely packed podcast episodes we've ever done. We named this episode The Lyme Rebel because a rebel is defined as a person who breaks rules that hold back themselves and others. And thank God this Lyme Rebel exists because this person is responsible for making more discoveries that have changed doctrine that was preventing you from getting the treatment that you needed and deserved than anyone else we've ever interviewed. This is the person that was responsible for discovering that ticks will spit in more than one bacteria into your system. Doctrine that had existed before research indicated that you would only get the Borrelia burgdorferi spit into you, and she was the person that has now demonstrated that Lyme disease is a polymicrobial infection. This is the person who's responsible for demonstrating that the Lyme bacteria, or the traditional Lyme bacteria, Borrelia burgdorferi, has the capacity to change its shape, to shape shift and go into a cystic form so that your immune system will not be able to kill it. And this is the person who is responsible for demonstrating that this bacteria will join with the other bacteria that was already in your body that you've been picking up for years and form a biofilm to protect themselves collectively from being killed by your immune system.
And Rich, more recently, Professor Sapi was begged by a group of nurses to investigate breast cancer and Lyme disease. And upon studying 400 breast tissue samples, she found that every single one of them had Borrelia burgdorferi and connected Lyme with breast cancer. Further research found that endometrial cells and ovarian cells pertaining to cancer all had Lyme disease as well. She also talks to us about how genetics in our human bodies combined with environmental factors like infections, aka Lyme disease, will trigger breast cancer and a wide variety of other conditions like autoimmune disease. Now, this can be really scary, and Professor Sapi provided hope to our listeners by providing solutions she found from her research to combat things like biofilm and also even things like breast cancer and Lyme disease, which she's currently working on as we speak.
So, folks, if you don't listen to any of the episodes that we've done in the past, literally any of the 350 episodes we've provided for you in the past, you can ignore all of them. And this is the podcast that we need you to listen to. You need to listen to this. You need to encourage everybody in the Lyme community to listen to this episode. And you need all of your friends who should be afraid of Lyme disease and they should be protecting themselves from getting bitten by ticks. They need to listen to this episode. So without further ado, we're really excited to introduce to you the Lyme Rebel. Hello, Professor Sapi, and welcome to the Tick Bootcamp podcast.
Thank you, Matt. Thank you, Rick, for having me. And I hope I have all the answers you're looking for today.
Well, we are sure you're going to have all the answers that we're looking for. And, you know, our community knows that Matt and I are two of the biggest Lyme geeks in the world. And we shared with you offline that having you on this podcast is like a birthday present to the Lyme geeks. So we're hoping our excitement doesn't boil over and we get too excited when we're asking you questions today.
All right, thank you again, and thanks for this great introduction.
So, Professor, why don't you talk to us about your background? I detect an accent. So why don't you share with folks where you began your early life?
Okay, so I'm from Hungary, and I got my PhD in genetics back in long time ago. And after I joined as a postdoctoral researcher at Yale University when I was doing cancer research for 15 years. And I've always wanted to teach. So I was applying for a teaching position for a local university, which I got accepted. And I wanted to teach and do my research, and all my research was cancer related. And but somehow I couldn't get my grants, and even worse, I like to hike. So you know what happened? I got Lyme disease. So it was a rough 2 years. I just started my professor position at University of New Haven. And I was scared. I couldn't really teach. You know, I was extremely dizzy. All the symptoms which you all very familiar with, I had. And doctor to doctor, And unfortunately, I couldn't get answers. And I was so ready to get this new position, and you could imagine how I felt. And being a professor, obviously, the first thing you do is always what's going on here. So I checked the literature. What do we know about, this kind of condition? And Lyme disease showed up. And of course, knowing that I'm hiking, and actually I had tick bite. And so I asked for a test. It was negative. I asked for all kind of other, potential answers, and it wasn't coming to me. So at one point, I was so dizzy, they offered some MRI, and that shows some very interesting changes in my brain. And the MRI doctor said, this looks like a Lyme case. And that's how I start to finally get some kind of treatment. But it's a long journey, another 2 years, and struggling to get better. And of course, as I mentioned, I wanted to know what's going on with Lyme. Why just not have my 2 weeks of doxy and I'm fine? Yes, and reading the literature, it was very clear to me that we don't know not much about Lyme disease and how to really treat this ancient bug. Borrelia was on this planet for a long time. So at this point, obviously, my research is not going too well. You could imagine I have no money, but I said, all right, I know some molecular techniques. I want to know what is in the ticks. So the next couple of years, I was in the woods. I wasn't scared at this point because now I'm going after you. Okay, you find me, but now I'm finding you. And we collected a lot of ticks. And the good news about collecting ticks is it doesn't cost money. You go to the woods and have your just a little canvas and you can collect. We collected over 5,000-6,000 ticks, and we tested them. So we found Borrelia. That was easy. But we found also all kind of other co-infections. And at this point, I said, okay, I have data, I can publish again. And nobody believed us. They said our numbers are too high. Nobody believed that Bartonella can be in ticks. Now we know, but at this point, it was like a very shocking result. And at this point, I was start to get a little money, not too much, just a little money, so I can start to culture Borrelia, the Lyme bacteria, and start to sort of look at it, understand it. And that's when I, again, read the literature. I knew about the different forms of Borrelia, so I was looking for that. I was treating them with all kind of antibiotics in a test tube. I want to see what's happening at this point with those different forms. And that's how we discovered the biofilm forms. And that was so exciting, that because this, a biofilm form is the most resistant form. It could explain everything, could explain those long-term symptoms in some patients. So we started to work on biofilm. First of all, we had to prove that Borrelia can be biofilm, and that took some time. I think it was 8 thesis students who worked on it. And at this point, I started to have a bigger lab. Usually I have 8 to 10 thesis students working with me. So it's a big group relatively. And the last now almost more than 15 years, I trained more than 100 students in Lyme disease research.
Wow,
so
that's really cool.
So I always say, one person can do so much, yes. But when you train those students, you can do a lot. You're cloning yourself. And some of them actually did stay in their field. So I was very proud of them. And so back to the biofilm research. So that obviously we tried to find some antibiotics, antimicrobials to eliminate this form. It was very frustrating for years and years, because everything we found, it was always some leftover material, which of course it came back. I'm sure you familiar with we find some alternative agents. First some peruvian herbs. After stevia came, suddenly people was like, what? Stevia? Are you kidding me? We also work with bee venom. which worked very well, but I always tell people it's venom. Okay, so careful with that. And right now, the recent research, of course, is keep continuing to to understanding this biofilm form. We did a lot of tissue work. So, for example, we published a case with Dr. Ligner and Dr. Goldman about a woman who had Lyme disease and treated for 16 years, 16 years with different antibiotics, very, very aggressively. And the minute she had some insurance problem and she couldn't get more antibiotics, she developed very devastating symptoms and actually died. And the silver line was that we had access to her autopsy tissue. So that was one of the very important case when we knew this person had Lyme disease. We knew that, that is proven. It was culture positive, CDC positive. It was positive. So we understood that it is not like a case when we're not sure about it.
Beyond denial. No one could deny that she had Lyme disease.
Yeah. So, and we published a paper in 2019, and we found spiral heat and biofilm forms in every organs we studied. So that was shocking. We published some other papers on skin tissues, which was very important because that's when we established ourselves to see how Borrelia looks like in actual tissues. That was super important. Right now, actually, we started again. I started a new project, but it was based on an old project. So we had some access to some mouse tissue which treated with infected with Borrelia, and we asked the same question: what happened in the heart? What happened in the brain? What happened in the kidney? Can we find those biofilm forms? So just before I logged into this podcast, I actually was working on some images in the heart and brain when we actually see this very big, very massive biofilms in this mouse tissue. Right now, the next step is to see how the biofilm affecting the immune system, what kind of inflammatory markers it can trigger. So that's going on right now. And the latest is I don't know if you heard about it, that we asked the question. Remember, I have cancer research background, whether Borrelia indeed can be found in cancer. Now, I know it sounds a little out there, but Borrelia was indeed found in some lymphomas previously. So it was some anecdotal evidence that some women who had Lyme disease actually developed breast cancer very short after. So I was asked, the nurses it was in Prague, it was in nurses, and they asked me to please look into it. Please stain some breast cancer tissues for Borrelia if you can find it. So I actually purchased some commercial slides. You can purchase commercial breast cancer tissue slides, and it showed up very early. So this project is very actively growing right now. We purchased a lot of slides, and we already have some statistical analysis, and looks like it definitely can be found in breast cancer tissues. The latest one, actually, we looked at some ovarian and endometrial tissues. Again, we found them. So this is very, very new. We tried to finish it. In the meantime, bacterial infections and cancer connection is a very hot topic right now in our field. So I think that's also maybe can give more, information to those women who struggling with all kind of, not just with the classical Lyme disease symptoms, but they have issues with some hormonal issues, and we still don't know whether they are more risk for cancer.
And the cancer question is one that's very triggering to folks in our community. We've had some discussions with Dr. McDonald, for example, on this topic, and I can tell you that the folks in the community who follow us were very anxious about that, and many of them said, "Oh, it's bad enough that I have Lyme disease. Now I have to worry about I'm getting cancer too." So these topics are really important for us to talk about, so that folks can be prepared to determine what the best course of treatment would be, depending on how their disease or diseases are presenting. But let's walk all the way back, because you've given us sort of this overview of the brilliant work that you've been doing, and it was really exciting to hear the overview. But now we want to break it down a little bit. Matt and I are going to talk with you together about some of these things. So you did some of your early research and studies here, actually on Long Island, where Matt and I are located. I know that you spent a fair amount of time on Long Island, and then of course you went across the pond and you were working in Connecticut. And I'm wondering, with somebody with your background, what is it that you knew about Lyme and ticks and tick diseases, and what steps were you taking to protect yourself from getting exposed to these diseases?
Of course, I mean, again, I mentioned that that we collect a lot of ticks. So when you're collecting ticks, the first thing you have to protect yourself. Yes, you don't want to get bitten. So we purchased those white suits, okay, with the duct tape, and we looked like some aliens when we went to the woods.
Yeah.
But seriously, I do still like hike with my family. So we have a very strict rules. So we obviously long pants, the white socks, because you can see the ticks. The body is covered. We do use some, I'm not a fan of DEET, but we use some alternative, more like essential based protection. They smell very nice actually. And you have to apply it more frequently than some other product, but we found it very effective. But it's even that when we get home in a garage, we take everything off and goes to the washer and dryer, and we take all showers and wash our hair. So we take it extremely seriously.
Right.
And
so Professor, you're making sure that you are enjoying the outdoors. So you said that you're in the woods with your students collecting ticks, and early on you collected a huge number of ticks, 5,000 or 6,000 ticks that you were testing, and we're going to talk a little bit about that process. But even though it is potentially dangerous for you to be in the woods hiking with your family, you're still doing it, and you're still enjoying the outside and still getting the benefit of being outside, because that there are steps that you can take to protect yourself. So one of the things that you can do, of course, is that you can dress properly, and you don't have to be an alien every time you go into the woods, but you can dress properly and make sure that you're putting yourself in a position where you can see where the ticks are if they do ultimately grab you, right? The second thing you said you're doing is that you're putting on different forms of protection, some alternative protection, which you're finding to be helpful. But the next thing you're doing is you're taking all your clothes off and you're putting them in the dryer, and you're doing tick checks, right? You're checking yourself very carefully, and you're showering so that now we know, of course, you can't wash ticks off if they're already biting you, but if they're not yet biting you, you can wash them off, right? Mm-hmm. So there are a number of different steps that you can take so that you can continue to enjoy the outdoors despite being in a tick endemic community.
Actually, it was a Yale study who actually looked at whether the showering would help right after hiking, and actually it was a positive study. So that's why we shower. So we follow suggestions.
Yeah, and of course, so long as you're not being bitten, the shower is going to be very helpful. And of course, putting your clothing into the dryer is helpful, because we know that ticks need moisture to survive. And if you're able to whisk away the moisture from the tick, it will die, right? So two of the principles that we know about ticks is that they need blood and moisture. And if you can take away the blood long enough, they won't live. Although there are some studies that are suggesting that you live a long time without blood.
Yeah, I've seen a study. Yep.
Yeah, and of course, if you're whisking away moisture. So these are some of the great tools that we can use. Now, do you have any suggestions for tick checks, especially like partner checks? Do you engage in partners? So, for example, when you're out hiking with your family, do you have someone check you after you come in, and do you check other people so that you have the additional protection of having the eyes of someone else checking you and the hands of someone else checking you?
Absolutely, because your back you cannot check, is no way, obviously. But again, some areas like behind your ear, behind your knees. So certain areas actually ticks actually love. So and if it's a nymph bite, you cannot really feel it. So not like, okay, I just check myself. No, you really have to have some help from somebody. Depends again on a hike. If it's just a very short hike, maybe just a quick shower. But if it's a long hike, you went like for a whole day, absolutely, you have to.
Okay. So one of the things that people can't see, because this is an audio only podcast, is you were actually checking yourself while we were talking about it, right? And what you're doing is you are rubbing your fingers across your body, right? It's because our fingers are very sensitive. In fact, they're the second most sensitive sense organ actually of any animal in the world. And because our fingertips are so sensitive and they're such powerful sense organs, it's actually your fingertips rubbing it through your body, all over your body, is what's going to find the tick more likely than you being able to see it. Because as you said, especially with these baby or nymph ticks, they're so small, they're almost impossible for us to see. Very
difficult to see. And don't forget the larva. Unfortunately, we had some, I stepped into a nest, and we were wearing the suit, the white suit, and suddenly I just was covered. So obviously we ran out immediately, and actually it was, I asked everybody just help me to take it off. And of course that was a little scary, I have to be honest.
Yeah, no, that would scare the bejesus out of me too, boy. If I had all those ticks all over me. So that must have been a scary moment. So let's now talk about some of your early research. So you have this journey with Lyme disease yourself, and it takes you almost 2 years to get diagnosed. And of course, that's a scary concept. I mean, you're a professor. You are studying at some of the top universities on the East Coast. You are now running, a department at, one of the top universities on the East Coast, and you can't get diagnosed. And you have a background in medical research, and you're struggling to diagnose yourself. So talk about why you think there were so many challenges for someone like you, who has as many resources as anyone. Why was it so difficult for you to get diagnosed?
I want to say something. I remember those times, and I remember I was doing cancer research at Yale, and I was doing breast and ovarian cancer. Ovarian cancer, one of the deadliest cancer, unfortunately. So anyway, so when I cannot get up from the chair, I was so dizzy, I couldn't function, and nobody can find anything. And I almost compared to myself, if I would have an ovarian cancer, at least they would know what I have. Not knowing for such a long time was so devastating. And now looking back, I know that not everybody making enough antibody. Some people needs more times. I'm sure you heard that some people months and months when they make enough antibody to show up on a test. We know there's now multiple species, which we didn't know, and some species doesn't show up on a test, and the bullseyes are not always present. So this is and one more, the symptoms sometimes is not that obvious. Not everybody having just arthritis symptoms. I heard the weirdest symptoms with Lyme disease. So having these very obscure symptoms, that test is actually not good enough to find, maybe a low level of antibody. And the fact that we have multiple species, I'm pretty sure you heard about that some species doesn't show up on our Western blot, because a Western blot was based on one species of Borrelia, one species.
Right. So let's hold on to that for a second and bookmark this, because it's important to recognize that there are challenges with getting diagnosed clinically, and there are challenges with getting diagnosed with diagnostic tests. So let's focus on the clinical pieces first, because you're spending your time on Long Island and in Connecticut, the places where Lyme disease was first discovered as an observational event in Lyme, Connecticut, by, interestingly, a rheumatologist from Yale University, where you were doing your research, and the Borrelia was allegedly, and I'm going to leave alleged out there for a moment, was allegedly discovered when a tick was dragged on Long Island from a professor at Stony Brook University another place where you studied. So, I mean, I don't. The irony is certainly not lost on me that that you were doing your research at Stony Brook and at Yale, and these are the two universities where the observational data came to define the term Lyme disease, and the bacteria was ultimately discovered in a tick that was dragged by a professor from Stony Brook University. Yet you're sick, showing classic symptoms of Lyme disease, and you have difficulty even getting a clinical diagnosis, despite being at the places where this disease was discovered in the 70s and the early 80s. So give me your reaction to how you feel about that, not only as somebody who is very resourceful as a professor and has all of the opportunities to get the best healthcare in the world, but also the irony of you studying at the two places where this disease was first discovered, and you weren't able to get a clinical diagnosis.
Yes, the irony wasn't really, didn't feel good at this point. I have to be honest. I felt like totally hopeless, and I felt like as a lot of other Lyme patients, is it's all in my head, and probably I have something else. Searching for answers. No, it was a rough time, and in the last actually it's practically 20 years now, I met so many Lyme patients. So we always discussing our journey, during those periods, and some of them feeling better, and some of them not. Even some of them have the resources to explore different treatment options, and still don't feel better. So that's frustrating a researcher to, as you can imagine. So
yeah,
sometimes it's difficult to look back to this time, because I know how difficult was it, and every time I get a phone call, those feelings always coming back to me.
I'm sorry, and I'm sorry we're taking you through this, but it is really important for us to sort of identify these issues so that folks can be validated on their journey, because so many folks have had these challenges with getting a diagnosis, and family members not believing them, and friends not believing them, and doctors not believing them. And it's important for them to know that if it could happen to you, it certainly could happen to the rest of us, right? Between your education, your background, your training, your contacts, your work as a medical researcher, if you could be gaslit that way, and you have such a long journey, it certainly makes us feel better that it can happen to the rest of us. Not that it should ever happen to anyone, right? So let's focus a little bit on the challenges with getting a diagnostic test, because we've now defined the challenges with getting a clinical test. And let's go back to the tick, right? So we know that ticks are the preferred vehicle for Lyme disease to be transmitted, right? And there are a couple of reasons why that happens, right? And Matt and I talk about this all the time, and Matt's going to start to come in and share with us, right? But one of the things we know about ticks is that there is a protein exchange, or there are some exchanges that happen between the bacteria when it's in the tick's gut and the tick itself. And what the research is showing is that when a tick spits the Borrelia bacteria into us, that bacteria is now supercharged, because it has the Borrelia has now proteins that it's been transferred from the tick, and now it becomes like a more powerful swimmer and a more powerful bacteria. So talk about that piece of this, and this sort of symbiotic relationship that's developed between ticks and these various microbes, and we'll talk about Borrelia first.
It's a very interesting point, because there was a study. They want to know how fast Borrelia can grow when the blood meal comes in, because some idea it has to go through the salivary gland, and that's how gets into the tick bite. My question was, okay, so when you have a straw and you're drinking your lemonade or whatever you're drinking, the straw, there is a backlash, yes. So my question was,
backwash, yeah.
Backwash, yeah. So my question was, why it has to go through the salivary gland? I actually discussed it with Dr. McDonald several times. Because Because if you look at the data from the salivary gland, it looks like it's only a few spirochete, few Borrelia can get into the skin. And I said, yeah, but I mean, how about what's happening with the regurgitation there? Yes. Is it possible that maybe it's not just salivary glands? So that's somehow these people, the scientists, they didn't really answer this question. The other thing is, when you grow, Borrelia is growing very slowly in the lab. I mean, we have a very, very heavy, rich media to feed Borrelia. It's still very slow. My student just sent me some pictures from, we have some new culture, and it's like a few here, a few here. But when the tick bites and getting the blood, the growth rate is unbelievable. So between within a very short period of time, it is like thousands of spirochete appear suddenly. So it's definitely an amazing relationship between the tick environment, and Borrelia. So obviously they evolve together. They understand each other. But again, I think even that part needs to be studied more. The other thing is, you mentioned about the protein changes. Borrelia can change everything within no time. I mean, you know, the cis form, how the cis form forms within seconds, for example, and it can, this whole outer membrane antigens. It's amazing how fast it can change and hide from the immune system or become a supercharged bug. So again, this bacteria is on this planet for a long time, knows what to do.
Professor, I did not know that when the Borrelia goes from the tick and the salivary glands to the blood, that the quantity or the concentration increases drastically. So that was really interesting how you described that. Thank you for sharing that with us. We've never heard that on this podcast before. So one of the things that I know every single listener to this podcast is dying to know, what did the brilliant professor do to treat her Lyme disease once she was diagnosed, right? When you got your diagnosis, now you're in the know, you're in this community with all the experts. What kind of treatment did you get to treat your debilitating Lyme disease that you realized you had? The
first one was just a short term of antibiotic, which just didn't do anything. And after, I was searching for answers, and I found some alternative treatments. At this point, you know, the Samento-Bentelol, I'm sure you heard about those Peruvian herbs. I was on the whole, Cardin protocol for a long time. People always asking me, oh, so should I just have like a month? I said, I took me like 2 years, literally 2 years to, but at this point, you know what, you can give me like bee venom or any kind of venom, I would have taken it. I wanted to get better so badly. Yeah? So to me, one thing about alternative, treatments, that I felt like I'm in control. Because when you go to the doctor and doctor gives you 2 weeks of doxy, and after you're done and you go back, I still don't feel good. And if you're lucky, maybe you get another 2 weeks of doxy, yeah, if you're lucky. But that's, you're done. After you're just done. And after you can run, million other doctors to find maybe, okay, I don't have Lyme disease anymore because I got the treatment. So with the alternative therapies, I felt that I controlling the situation, and it really was very important for me at this point.
So you were quickly empowered as a patient when you realized the short course of antibiotics didn't help you, but you knew this was still there, and you decided to go to the Cowden protocol pretty quickly, it sounds like. So Samento, Banderol, and all of the other products are part of the Cowden protocol?
Mm-hmm, mm-hmm. I at first I just took some of the herbs, but I felt like a month into it, I felt like it's something changing. And when you feel something, you get excited, and I said, okay, just give it to me, and I can take all day, just let me feel better. And I did a lot at this point. Of course, you eat clean. I gently exercised. I didn't push my body. So it's very important. I juiced a lot. I made a lot of good Hungarian food, Hungarian soups. And I think one thing is very important, and people ask me what was the most, just believe that you can recover. I think that was very important for me.
So while you're doing all this, are your peers and colleagues shaming you or making you feel like kind of crazy? Because I would say most people that are professors or doctors sort of look down on holistic medicine and herbal medicine, right? So what was the feedback you were getting from your peers, since you were this academic and you were leading these research studies in the cancer community and in the Lyme community?
That didn't go too well, obviously. At this point, I remember I don't know why I Googled myself, and you shouldn't not do that. And I remember one post is like, oh, she was such a good researcher at Yale. I don't know what happened to her. Now she's doing this crazy research. So that was the feedback. But again, especially after I really felt like I'm feeling much better with the alternatives, that's just confirming that is something there.
Yeah, and little did they know that your work in cancer research and your work in Lyme research would be brought together, which we're going to talk about in a little bit. And it was really exciting to read that research on the internet about all the work you're doing. But I do want to talk more about your progression here, right? Because now you're starting to pivot from cancer research to now, studying ticks. And it sounds like you said earlier with Rich that you started to identify that Bartonella can be transmitted from a tick and infect a human. But at the time, people didn't believe ticks could infect humans with Bartonella. And you were the one who said, hey, guys, there's something here. And you brought that to light. So is that what we heard earlier?
Yeah, they didn't even believe that Bartonella can be in the ticks. But it wasn't just Bartonella. We ran a lot of different, at this point, not much money. My budget was ridiculously low. So I only can do one kind of experiments, collecting the samples and what kind of experiment. But with this limited money, we still found a lot of different species. Probably heard of it, Mycoplasma, Ehrlichia, of course, Bartonella, Babesia. We found the species, and one of the study, which again nobody wanted to really acknowledge, that we found multiple species in one tick. And it's now the studies are coming out, and now it's more accepted. At this point, they were like, no, your number's too high. It is all just some kind of lab art. I don't know what you're doing in the lab. Everything positive. No, wasn't everything positive, and actually we had very, very good negative controls. But again, I felt like nobody wants to hear this. That's how I felt. Nobody wants to hear that this ticks in Connecticut, for example, is loaded with Borrelia and loaded with other bugs.
So, of course, that is scary, but also it makes sense that many of the researchers didn't want to acknowledge what you are finding in your lab, and that is that Lyme disease is not a single bug. It's a whole bunch of bugs, right? I mean, it really is a polymicrobial infection. And because the researchers who initially had discovered this disease in this vector in ticks had identified just one bacteria, you had to move the needle, and you had to move the research community away from what everyone believed at that time, which was there was only one bacteria in the bug. You're finding now multiple bacteria. And one of the things that Matt and I wanted to talk to you about today is that we have come across some research where there was a professor who had discovered that a tick could have up to 200 different bacteria, viruses, and protozoa in it. And we've only identified 18 now. Now, of course, when you were doing your research, when you first started, and that's what an unbelievable trendsetter you are fighting against whether or not there was just one. Now you show the world that it's more than one, that it's multiple, and now, as it turns out, what we're hearing is, well, there are only 18 bugs that can be inside of a tick. So I'm wondering if you can share with our community that one of the reasons why people are getting so sick, and one of the reasons why it's so difficult to diagnose, coming back to the the diagnostic part of this with the test, is that there are many bugs that could be in a tick, many of which have not even been discovered, and many strains of each one of these types of bugs can be inside the tick.
I mean, it's coming from a technical approach because we were trained that you check for one thing at one time. And the good news is now we have better understanding that in a bacterial, viral, fungal community, they are interaction. So they live together. So it's not just bacteria, not just fungus, not just viruses. And now we have the technology to check that. So we have, for example, we can sequence, the 16S ribosome RNA, which, for example, going after bacteria, we can sequence everything what is in the ticks. And I know the studies are coming out. It's absolutely beautiful, and I'm loving them. So again, maybe it was a little bit of technology, or maybe just our mindset that needs to be changed. And 5-10 years usually when you able to make those kind of changes in the science field, unfortunately. But 5-10 years for a human is a long time. Yes,
it is a long time, especially when people are really sick. I mean, asking people to wait 10 years before they can take action in order to heal is just a terribly long period of time. And in many cases, folks can suffer, damage that may be irreversible if they're not able to take, earlier intervention. So it's a lifetime for many people if we have to wait 10 years. And that's one of the things that we find to be, very frustrating as folks who are, following this.
Rich, I just want to jump in and just make a comment on that, because although it's very frustrating, and as somebody whose life was, significantly changed because of Lyme disease, I just want to thank you, Professor, because if it weren't for your persistence and hard work, we would be even further behind in research. And I don't know that I would be where I am today with my health if it weren't for your hard work and research and advocating for us in the Lyme community. So I just want to recognize that piece of it and thank you for your hard work and persistence, because yes, it's frustrating we're not further along, but we wouldn't be where we are if it weren't for you. So thank you.
Absolutely. No, thank you. I want to join Matt in thanking you, because if it weren't for people like you who are willing to buck the system, you had no money. You had very little support from the standpoint of having staffing, but you as first a patient and ultimately as a researcher were willing to buck the trend, and you were willing to say, no, all of you Ivy Leaguers are wrong. Mm-hmm. It isn't just one bacteria. It isn't just one bug. It's more. And you literally changed the world here. So I want to thank you for that as well. It's really, people would not be healing at the rate they were if it weren't for you. So thank you for being brave, and thank you for being willing to stand up against, what was the standard belief.
Yeah, I mean, I can tell you some stories what happened. They want to shut down my lab, for example, very, very, very badly, very badly. At this point, I had a very open-minded and very science-oriented chair who getting emails after emails that lab has to be shut down right now. And he just said, no, she's maybe a black sheep, but we need black sheep in science, because we cannot just always go in one direction. We need to check out some other alternative ideas. The biofilm idea also was extremely controversial. I got emails from established scientists that I never seen biofilm in let's say in infected mice. I said, did you look? And I said, can I look? Would you mind to give me some samples? I'm more than happy. I don't want anything. Just give me some samples, some leftover. You infect the mice with Borrelia. Give me some tissues. They never did. I kept asking. But finally, one researcher said, I helped you with that. That's why I had some samples. So that's why actually the mouse data never got really published, because it was always on the back, because I didn't have enough tissues. That's why actually the human data got published first. And it's thanks for the Austrian dermatologist who said, I want you to check this out, and I send you some samples. So I find help all the way, but most of the time I find a lot of resistance.
Yeah, but Professor, I'm curious, how did you stumble upon the biofilm concept, right? Because you're the first person that brought this to light. And this is another huge concept that prevents people from healing and people need to know about. And you discovered it, you found it. So can you walk us through that aha moment when you realized, wow, there are biofilms and they make treating Lyme disease far harder? We'd love to hear that discovery moment you had.
So at this point, back in that, first in the woods, collecting ticks and doing some simple analysis for what could be in the ticks, I really want to see Borrelia. So I found a very old microscope but it worked. And of course, we got some culture. At the beginning, we couldn't even culture it because it's turned out a little bit more than to just put it in a culture media. But we figured it out, and I said, okay, now let's see what happens if I add penicillin or doxycycline. What happens to this bug? Can I kill it? And so we started to add, in the culture tube different antibiotics, and again, this is a very simple microscope, but we were able to see those morphological changes which I read about it, and I was very, very impressed by that. I remember I want to make the cyst form. So Bronson is a scientist from Europe, and I emailed him. I said, how can I make cyst form? I want to make cyst form but quickly.
Professor, can I interrupt real quick? Can you, for our listeners, explain, we know you're an expert in the morphology or the different shapes of the Lyme bacteria. Can you explain for our listeners the cyst form versus spirochete form, just so we have a general idea of what they are?
So Borrelia has lots of different forms. The main forms are the spirochete, which is actually beautiful. I know that sounds a little crazy, but under the microscope, if you see it, you will be like, what? And what early research, again, research from Europe, found that if you start to add antibiotic, but even not even antibiotic, any kind of manipulation with the culture, Borrelia within like 10 seconds crawl into this very like round kind of bodies. It is amazingly fast. If you want to watch it, go to YouTube and Google Borrelia and I guess cyst form or round body. There's a video which I always show on my conferences because I love the videos. So it shows how quickly it responds to the environment. It's amazing. So that's and if you look at the culture, you see the beautiful spirochete form, but always little specks next to it. And I never understood that. I thought it's maybe dirt, who knows what is it. Yeah. And we had a very special strain of Borrelia, which is green fluorescent. So when I looked at under the microscope, this green fluorescent Borrelia, I see the dirt. So it is Borrelia. So this form is actually in the culture all the time. I always call it as a backup form. This is like, you make a backup for your computer. This is made a backup for whatever happens, if something happens in the environment, we have this population who is gonna survive it because it's protected and able to come back one point. So that was one other question. So, okay, so I induced the cyst form, which I learned how to do it. And I said, how long I have to wait to come back?
Back to spirochete, right?
Yes, so the spirochete turns into this round body, so cyst. And again, I talked to this European scientist and said, just change the media, remove whatever you put in and check every week, but I promise you it's gonna come back in 3 weeks. It was 21 days exactly. I couldn't believe it. And I see spirochete again. So it was like, what? How do you know that exactly 3 weeks? I guess he probably did a lot of experiments determine this time frame. So that's one. Now, there's also other forms, and we don't really understand the other forms. They're called the cell deficient forms, for example, which is, we need to learn more about it. And when you look at under the microscope and you start to grow Borrelia and let it grow, and when it's getting confluent, you start to see that they're gathering. So the spirochetes start to find the little space together. And that's called the aggregate forms. And at this point, when I looked at the aggregate forms, I said, what you guys doing there? Are you guys meeting? Planning some kind of,
some evil plot.
evil plot. So, and I was at this point, of course, I'm reading literature very heavily and looking the pictures of these aggregate forms and what I'm seeing, in literature. And I said, there's such a thing as biofilm. And I wonder you guys making biofilm here. And at this point, Dr. McDonald and I sort of sharing information. So he sent me pictures. I look at what I found today. There was one, that was when we published it. There was one picture, so 2 aggregates, big aggregates, and it's like a bridge between them. And that was the aha moment because I said, they're talking. they are talking. And I only know one form which can talk is biofilm form, because the whole talking and quorum sensing, this is, this is what they do. They, like, quorum sensing is you texting each other, okay? That's how I explain to my students.
That's insane.
It is insane, Matt. So let's bring this back to why it is difficult to diagnose this disease, and then why it's difficult to treat this disease, right? Because one of the reasons why it's difficult to diagnose this disease is because with traditional testing, they're looking for antibodies, right?
Yes.
But of course, this very sophisticated bug goes through various forms of shape shifting, so that your immune system is not able to respond to it. And because your immune system is not able to respond to it, there are not going to be any antibodies to test, even though this bug is still in your system, which 21 days from now, it'll go back to being what it was before, and reproducing and attacking.
It is mind blowing, yeah. And it's even more because I read at this point, we didn't have like SARS, we infected like mammalian SARS, like, let's say right now we're using this breast cancer SARS, we're infecting with Borrelia. But I read like some fibroblasts, which is skin SARS, can be infected by Borrelia, but I never really seen a good image of it. And one of the things really wanted to know whether Borrelia can go inside the cells, because we always talk about it's gonna just maybe make biofilm in the collagen-rich tissues, and some spiral gonna come out, go to and travel somewhere else, make another biofilm. But I said, can you go inside the tissues? Because that would be interesting. So there was few papers suggesting it. So we had two different cell type. One was a neuron cell type, and one was the breast cancer cell type. And we both we were able to prove that they can actually go inside. If you look at the latest paper, we used a very special microscopy techniques which can sort of look inside the cells, and we found Borrelia right there. And what is very interesting, because I heard something called the intracellular biofilm, which means biofilm inside the cells. I said, no way, no, it cannot be, no. And I'm looking at this microscope images, and I can see similar aggregates forming inside the cells. So this is the multiple layer of craziness. Yes.
This is creativity,
nuts.
right? I mean, of this bug. So you've now taught us that the bacteria has a relationship with the ticks, right? And there is exchanges of protein between the tick and the bacteria. This now supercharged bacteria gets sent into our system. It gets into our system, and it goes through these various processes of shape shifting so that our immune system cannot kill it.
Well, before that, when it hits the blood, it increases in quantity significantly. We learned also from Professor.
Right, increases in quantity significantly when it comes out of well, whether it's in the salivary gland or not, that's some question, right? Yeah. It now goes through this process of shape shifting. But there's also this relationship between not only between the Borrelia themselves, and they're coming together in this biofilm, and I want to hold on to that for a second, but they also have a relationship with these other bugs that you were the first to argue and then ultimately prove that are being spit into us. So these other bugs are working on us and helping to keep each other from being killed by our immune system, which is now responding to all this. And now they come together and they are forming a biofilm. Can you describe what a biofilm is? And can you also describe for us whether or not it's just Borrelia that's in the biofilm, or are there other bugs inside the biofilm? And what impact does the multiplicity of bugs inside the biofilm have on what's happening with those bugs inside the biofilm, in addition to protecting it from the immune system killing it?
So think about biofilm is like a city. What we do in city, we protect maybe a city wall or something, yes? But also very importantly, in a city, you have to share, shared task, yes? So you have to have different population doing different tasks. You also have to bring in food, and you have to, expel the waste. So there's a lot of things going on in a city. So when we looked at, we had a very fancy microscope at this point. We were able to look at how the city is built by Borrelia, which was absolutely beautiful. they're like a crochet kind of structure forming. And we started to see, we looked at every day, we started to see little holes in the structure, which of course made sense because that the holes can be used as a channels, yes, to bring in food and expel the waste. But we also seen multiple layer protection. So this is the wall covering this biofilm, and this is it was known from some other biofilms. They are slimy. They call them mucopolysaccharides, like sugar kind of sliminess. And when I talk about biofilm, I said, you know, biofilm, how about you just don't clean your shower for maybe a week? Get it slimy? Oh yes, and you know what you do? You have to take the bleach and the brush and brush it off. It's not going away very easily. Or, you know, a lot of biofilm in our mouths, unfortunately, you know, and that's why we have to go very high, you know, good hygiene. And
brush your teeth.
You brush your teeth, but unfortunately biofilms also found in, you know, those some kind of implants, which I have a family member, unfortunately, who got a knee replacement and got an infection. They could not cure it, and that was a biofilm kind of infection. And this is one of the things that we still have to understand how to deal with this biofilm. And back to the biofilm is very rarely monospecies. Monospecies means only one species. Usually it is multiple species. So we did, remember the skin biopsies I got from Austria? Yeah. So fortunately I had a lot of samples. So I able to do a lot of experiment, and I was able to see not just Borrelia in a biofilm form in the skin samples, but we started to look at other bugs, and we find this chlamydia species and H. pylori species, and the how they embed themselves into the Borrelia biofilm is super interesting. Like chlamydia, it's exactly in the middle. And almost feels like it's using, I call the iron castle, which is funny because Borrelia doesn't use iron, but chlamydia does. So I always wonder, the chlamydia is there to make the iron castle, because iron actually is one of the well-known material for other biofilms, but Borrelia doesn't use iron. So I said, oh, so you invited your friend here who's bringing the iron. Now you can make the iron castle, okay? And again, H. pylori story, we still don't understand exactly what H. pylori does in this biofilm, but obviously they're plotting some path, you know, to see how to survive the environment. How many more? I mean, we can just keep looking and looking and looking.
So, Professor, what I'm wondering is, you know, and again, this bacteria is very resourceful, right, in developing relationships with ticks, developing relationships with other types of microbes in the tick. And then when it comes into our body, it seems what you're saying is it's then gathering other types of microbes that are already in our system that didn't come from the tick, that our body had been managing as part of our microbiome, and they're now combining forces with these other microbes, which weren't dangerous to us before, but when they get into the biofilm together, they're now supporting one another and now supercharging each other, because they're now exchanging proteins, and they only have the ability to change their shapes, but now they have the ability to change their essence by having more proteins and essentially, I guess, evolving within this biofilm, so that ultimately, if the biofilm breaks down, it's a very different microbe, and the immune system may or may not have the ability to recognize this evolved now microbe.
This is super important to understand what exactly happens inside the biofilm. This synergetic relationship between these different bugs. I think that we need to grow appreciation for that. We always think about to kill one bug. Yes, no. I think especially when it comes to biofilm, I always look at Borrelia as a driver. I think is one of the most ancient bacteria on the planet. So I think that able to make again the iron castle, this is a perfect refuge for all those bugs. And probably they were swimming with joy, you know. Finally we find something which is such a structure that we can survive it for a long time. So I looked at other biofilms, you know, other structure. I don't see this kind of very almost like a architectural planning, how to make this whole environment for us that we all survive.
I just want to ask a question about, because beyond the severity of these biofilm, you taught us that biofilm can exist in the blood, and also we know from Dr. McDonald in the brain, but then you touched on, Professor, that it can go inside the cell. So not only did you teach us that Bartonella can be given from a tick, which nobody believed, not only did you discover biofilm, and now next you discovered that Lyme itself and biofilms can be intracellular inside the cells, which is something that you discovered as well, right? But what does that mean for us and how we can heal from Lyme and our overall health? Because if it goes inside the cell, is it likely that cell is going to die? And what impact is it having on the human body? And what impact is it having on the bugs to survive and thrive and make us even sicker?
Right. I know that a lot of you right now saying, oh my gosh, I don't know what's going to happen here. It is not inside of my brain, inside of my blood, inside of my cells. Again, we have to understand this bug first, okay? So I always say, understand your enemy. You cannot do anything until, okay? So knowing what it can do, it is extremely important, because maybe the antibiotics are not the answer here. Maybe we really have to rethink how we approach this even clinically. And there are, I'm pretty sure you heard some alternative, which is working totally different ways than, try to just kill the bug with some antimicrobials. So I think we have to, again, I would love to see more research. Again, those research are expensive because, I did a lot of test tube research, and I think that can take use at one point, but after you really have to look at this bug into an actual organism to understand. You see how many different places it can hide. So we need more research for that, and we need to see some other alternative ways to get this out from those hidden places, and after maybe hit it with some kind of strong antimicrobials.
But although we need more research, you partnered with Columbia University, and you mentioned this earlier, right? This person you were monitoring had Lyme and was treating for 16 years with antibiotics. And after the passing, you found that there was still Borrelia biofilm in the tissue, right? So, and your studies are happening, and they're being reinforced that antibiotics alone probably are not going to cure somebody of Lyme disease, because 16 years later, this person still had Borrelia biofilm in their bodies after passing, right?
Yes, I mean, obviously we still would be nice to have antibiotics. Yes, that would be a dream, no? You just want to go there. I remember I was presenting at ILETS, and I think it was a little controversial. It was very early in the morning. I said, I understand we all work in chronic Lyme, but would it be nice if the 2 weeks antibiotic would work for us? And I remember the face of the audience was like, what? I think I pushed the wrong button at this point. But I mean, seriously, it would be nice to find it. Now, we didn't give up on antibiotics, okay? So we're looking for antibiotics which might be able to penetrate inside the cells, penetrate to the brain barrier. It's still hope there, but at the same time, we have to start to look some alternatives on how to get them out from the cells, how to reach these hiding places. For biofilm and its collagen embedded, that would be a tough one. That will be one of the toughest. Collagen, that rich matrix embedded biofilm.
But you've done some studies, right? You keep talking about alternative medicine. You mentioned earlier this whole, and this one, when I first got Lyme, I remember hearing about the stevia extract as being a potential, treatment for biofilm and deep Lyme disease. And a lot of people telling me, oh, that's crazy talk. That's witchcraft, right? But you're telling us that there's some evidence behind this, that this is actually an effective way of breaking down the biofilm and treating people that have chronic Lyme. So can you walk us through that transition of your research of how you even discovered stevia, and what your findings were about how it worked and how effective it was at breaking down biofilm and helping patients that have chronic Lyme?
So the Trojan horse?
Yes.
Yes. Okay. So the question is, so we know that the biofilm is covered with lots of protective layer, but cannot be totally isolated because they need to eat. Yes. I mean, right. Most of the cells are sort of a dormant stage, but still, I mean, they need some food, and they have to expel the waste. So my question was at this point, first of all, how to wake up those dormant cells. That was very important for me, because if it's the dormant or antimicrobial, not gonna work. Antimicrobial working on growing cells. So that was my one question. I said, and how can we send it in? Okay, how can we get the antimicrobial inside it? Because if we cannot get inside, there's no hope for here. And that's when the Trojan horse comes. So, you know, the whole story that you hide it as something else. Yes. So the first question, remember, the first question was how to wake up those dormant cells. And I read a paper. It was a Nature paper that they add sugar.
And when you say dormant, do you mean the round body cells?
Round bodies and
cell deficient,
with all kind of different cells. Some people call the persister cells. Just inside the cells with a very low metabolic state. Okay, I call them the sleepers. Okay, right. So how can I wake up the sleepers? Because I don't wake up the sleepers, my antibody don't work. So it was a study that they add a little sugar, different kind of sugar. It was an E. coli, actually coli culture, not Borrelia, but same idea, how to kill it. And after that, the antibiotics. So the whole idea was wake them up with sugar, okay, and after hit them with some antimicrobial, and they said it worked, okay. So I got very excited, okay. I said, guys, we're gonna wake them up. And in this study, they use different kind of sugar. So I said to one of my students, please go to the grocery store and just pick up whatever you can. I have some sugar here in the lab, but whatever, everything, the fake sugar, everything, just bring it to the lab. So of course she bought whatever she found, you know, and stevia was one of them at this point. And we're doing the study, and the very first experiment looks like stevia is not just waking them up, they're killing them. I said, how could it be?
Wow.
This is a, you know, at this point I knew about stevia, like, you know, some put in my coffee, if, you know, to make it sweet. So I was reading about it as a Japanese leaf, and in Japan they use it for antimicrobial for centuries. So it's not, the Japanese people not just using it to make the tea sweet, but they take it as antimicrobials. I said,
any correlation to Japanese knotweed, or am I totally going off with that one?
I don't know, I'm not sure. And so I said, oh my gosh, is my working as a Trojan horse? Because Borrelia thinks it's food, and it's not just food, it's antimicrobial. So at this point we jumped on it, and we published it. We had a huge study. We compared it actually with other antimicrobials, which didn't work as well. So I know some of your audience probably using the stevia already in the protocol, and I again I don't have any clinical study. Dr. Horowitz has some study on it, and he's still using in his clinic. He believes that helping even with his antibiotics treatments. So right now stevia is included in the Cardon protocol next to some etobendarol. Actually, we just about further this study. I want to see if stevia would help to other antimicrobials, because I want to see whether it's true. But again, there's again test tube experiment, and as I mentioned, I really want to get out from the test tube. So we had this crazy idea that, you know, we cannot afford mice; it's very expensive. Of course, we can't use human for obvious reason. So we had a zebrafish facility on our department. So we actually did finish a project when we put Borrelia into zebrafish. So this is still under, because some issues, of course, is not that obvious, but we need some kind of model system when we can really study this bug in a natural environment, not just in a test tube.
Before we move on from the stevia, I just want to ask, because Rich and I always look for low-cost, low-risk options for people listening to this podcast who are looking for ways to improve their health and continue to fight tick-borne illnesses. So you saying somebody can go to the store, buy stevia extract, and take a certain amount of it, a tablespoon, et cetera, whatever it may be, and that can actually have a positive impact on their health?
Just for our study, again, our study is test tube study, so it's not a clinical study. We found that the powder doesn't work. We tried to dissolve the powder. It didn't have the same effect. So something about when they make the liquid form, so it's, I guess, some active ingredients in a liquid form. We test a lot of liquid forms. most of them worked. So you can practically go to the grocery stores. Again, this is a low cost option, okay? And you could try it and let's see how you feel. Okay, I never push any kind of, protocol on anybody.
No, but put it in your herbal tea that you're taking and put it in a cup of coffee, right? And you could potentially be helping yourself. If not, you're just getting a little sweetener in your coffee, your tea, right? I mean, it's a low cost, low risk tool to try, I feel like.
Japanese people believe in that they use stevia quite a bit.
So let's stay with this path that we're developing here, where we have this this very sophisticated bug, as you had said, this ancient bug that has existed on the earth long before we did, and has been going through this process of shape shifting and recruiting relationships with ticks, recruiting relationships with other bugs in the tick, recruiting relationships with bugs in our body so that it can go through the process of surviving. And let's talk about the antibiotic loop. One of the things that Matt and I talk about often is one of the challenges that we often see is someone takes antibiotics, they feel better, they go off antibiotics, they go back to the antibiotics again, and they feel better, and they go off the antibiotics, and they come back on them again, they go off. And what has us always anxious about that, Professor, is that at least, when Matt and I are talking about this, is of course, what's probably happening when we're going on and off of antibiotics is that the bugs are changing, right? Because we know that they're changing when they're in the tick. We know they're recruiting other bugs inside of us, and they're exchanging proteins. And ultimately, if you stay on that loop where you're just going on and off of antibiotics, it seems to me that what you're going to do is you're going to grow antibiotic resistant bugs in your system, and then the antibiotics aren't going to work at all. So what is your thought about the on again, off again relationship that many patients have with antibiotics?
Oh, I know so many patients who cannot stop taking antibiotics. Same vicious circle, yes. And seeing the case which we described, the 16-year-old treatment case, I never forget that I got just a few slides from Dr. Goldman just to check it out what's going on. And I just a few slides, this is an autopsy case. I didn't know that, what to expect. And it was a liver slide, and we stained it for Borrelia, and it is massive, huge, unbelievable structure shows up. To the point I didn't believe in it. I said, it's too big, it is fake. No, maybe we did something. So I said, okay, we're gonna repeat this in a different protocol. And we kept repeating it. It just keeps showing up. And after, of course, was some brain samples, some heart samples, and this extremely massive structure showing up. And again, I had just the skin samples before, which we had biofilm, but they were small, delicate. This was like a monster biofilm, which that's why I was almost, I couldn't even believe at the beginning. Fortunately, we had access to a lot of material, so I was able to keep doing different techniques, to be sure that what I'm seeing is real. So this patient took antibiotic for 16 years, on and off, same as most Lyme patients, and as you see, it's just practically grow this biofilm bigger and bigger and bigger. Now, I'm not blaming any physician or anything, because that's what a physician can do, just to work with the knowledge they have. And I know her physician was really passionate about her house. So I find, that, he was really trying with different antibiotics, whatever you can think of. And she was actually doing okay for 16 years. Not great, but okay, until, I told you some insurance problem and that cannot get antibiotics. So looking back to this case is really, we really have to appreciate, what this bug can do, and not just keep doing the same thing over and over and over again. Now, is the stevia will be the game changer? Maybe will be some other, maybe some interesting new antibiotics, maybe. maybe some alternative, I mean, probably heard about thrive therapy and some other therapies. We don't know yet. But again, people shy away from this kind of, alternative way of thinking. And I don't blame them. And they want to feel better. They feel better when they take antibiotics. But again, the vicious circle starts over and over again. Yes.
But is there a risk there, Professor, right? Because on one hand, we want symptom relief, but on the other hand, we don't want to kick the can down the curb. And I feel like antibiotics may be kicking that can down the curb because, to Richard's point, we're making the bugs smarter. They're becoming antibiotic resistant, and it's harder to get effective antibiotic treatment over time. So looking at this, from a zoomed out perspective, I feel like something that penetrates deeper, like a holistic approach, whether it's stevia, whether it's the Cowden protocol, and maybe it's a combination of antibiotics with those things, it'll get deeper. So these bugs hopefully will become, less and less and less, and not becoming supercharged to eventually make us even sicker in the long run, right? I mean, what are your thoughts on that?
It's interesting, we talked about microbiome, how important a healthy microbiome in the system. And we had somebody from UConn who was one of the experts in microbiome, and I was, of course, went to lecture. And after I was able to talk, I said, I'm doing Lyme disease research, and I was wondering how important a healthy microbiome, for a patient. And he looks at me and said, you know about fecal transplant?
Yes.
I heard about it, but of course I heard about it, but I didn't know too much, how it can be used. Said, I had a student who had Lyme disease and nothing worked. So, okay, that's going to sound a little gross. Okay, so everybody hang in there for a second. And she had a brother who is healthy. You know what she did? I
know where you're going. I'm afraid for you to say it.
I don't want to even say it, but you know what she did? And she said it worked. She felt much better. And I know that now some places you can actually, that's one of the treatment option. And I know it's not just for Lyme, but a lot of disease now is something which is start to get a little appreciation. Okay, they now is in pill form, so a little bit differently, so it's not as gross.
No, but Professor, you're right. I mean, we read an article in the last year that came out where chronic Lyme patients, they studied their gut microbiome, and they found a very high level of one microbe that isn't present in healthy people, and a very low concentration of another microbe that should be high in healthy people. And now they think they're finding a correlation between concentrations of microbes in the gut and chronic Lyme, because that has an impact on your immune system. That has an impact on your immune system's ability to go back and fight the microbes in your body, like Lyme disease, right? So I think there is a connection there, but we've barely scratched the surface to understand what actually is going on in our gut microbiome, which does represent 70% of our immune system, right?
Absolutely. And interestingly, we did do this kind of, microbiome analysis on the skin samples. Remember, we had the skin samples. And we sent it to this company who does the bioinformatics part, so understand what is exactly in the result. And the first thing they emailed me, and they said, it's amazing how little representation of normal bacteria in these samples, and the distribution between certain species was crazy. They didn't even say, where do you get these samples? Okay, so it's definitely something happening with the microbiome, and I think that's why when I say alternative has to be explored. This is the alternative. I would love to explore it more and more.
And this was the skin microbiome you're talking about. So this is you now found the skin microbiome to be drastically different than a healthy patient, and now we have other studies proving that the gut microbiome is drastically different. So obviously there's a connection there, right? And thank you. We haven't heard that piece about the skin component on this podcast.
Matt, can we stay for this one more second, because we're now making this connection between the use of antibiotics and the impact that it's having on your biome, right? We did explore with you, Professor, that using antibiotics over and over and over again is likely creating superbugs. But part of the reason why it may be creating superbugs is not only because the bugs are going into the either cystic form, or they're going into the biofilm, where they're now shifting with other microbes and changing their form, right? But ultimately, they just have to have one that meets the one reformation of the bug, And now the antibiotics are not going to work anymore, and it's just going to take off, right? But the other thing that's happening is that we're also changing our microbiome, and the antibiotics, because they're broad spectrum antibiotics, are in many cases changing our gut and what's happening in our gut. And now what Matt is just pointing out is part of the reason that may be happening is because of the tools we're using to try to kill the bugs. So we have this sort of multi-level chess going on here, where we're taking one step, we're causing the bugs to change, and now as a result of going into the biofilm, we're now impacting our gut microbiome, which is now having an impact on our immune system's capacity to succeed. And the cycle is increasing the likelihood that the bugs are going to survive, and we're not going to ultimately be able to beat this thing because we're overusing a form of treatment that over the long term probably is doing more harm than good.
I always say that, ultimately your immune system gonna cure this bug. Okay, we can't just have the immune system. Now, if your, as we mentioned, that our microbiome has a huge connection to our immune system. So my grandma, a little small village in Hungary, always said the most important thing is that you eat fermented vegetable every day, every single day. They didn't have the probiotics capsules or anything. They made in the kitchen, different kind of fermented veggies. So I grew up with that. So since today, and I think that's one of the reasons I was able to recover, because I was really was very big on that. My microbiome has even during the treatment. So people asking me what helped. I mean, I said, of course, the juicing, the Hungarian soups, and the fermented veggies. I did a lot of sauna, sweating. I said, so release all kind of junk from my system. Eat very healthy. Detox, it was very important to me. I had some supportive detox work, just be sure that my liver can handle all those detox. Yeah. But eventually, I think it was a very comprehensive approach. And again, the microbiome has to be there, has to be taken care of, because it's not going to work. Whether you using the fecal transplant or you are using the fermented veggies, but you have to think about that.
And I'm curious on your comment that the immune system alone will not get rid of the bug, right? Because we know at least 15% of the world population has Lyme based on that study that came out a few months ago. Many people have Lyme and go symptom-free. So they get acute Lyme and they move on. And now there's that debate, well, is their body managing it and it's still in their body but being suppressed? Or are they healthy enough where their immune system can actually eradicate the bug before it gets disseminated, right? So what are your views on that, Professor? Are you saying that the immune system alone cannot eradicate even Borrelia by itself, and that it's going to be dormant and just managed? Or do you think that the immune system could, and it's a combination of all the stuff from the tick bite that our immune system can't overcome on its own?
Yeah, unfortunately, there's a multi-layer question here, because we know that from the tick spit, there are some agents which can suppress our immune system. That's why you get in this big, huge, pool size. But at the same time, I remember I was walking on a campus, and I'm doing Lyme disease research, and this one guy coming, says, I got so many tick bites when I was growing up in Connecticut. I never got sick. And I was like, probably you had a very healthy immune system. And I think our immune system is not healthy anymore. That's why this pandemic right now, or I call it pandemic, some people epidemic, whatever. There's a lot of too many cases. And again, or treatment is not helping our immune system, or lifestyle not helping our immune system. Dr. Horowitz always said that he always has this big questionnaire for the patient. Said 90% of cases they come down with symptoms with some kind of tragic event in their life. And I was thinking at this point, so what was a tragic event in my life? And I was like, oh, of course, my mom died. Okay, so the whole stress and everything comes with the immune system is practically gone, and now the bug can flourish. Yeah, so but again, it's multi-layered, because the tick saliva have the immunosuppressive agent in it. Borrelia actually has certain ways to trick our immune system. When you have a test, they're checking for IgM and IgG. These are two antibodies.
Yes.
Yep.
The IgM should at the beginning when you get the infection, and should go down, and IgG comes up. Number one questions I remember in Lyme conferences: why do I see IgM, the early one, still in a patient who are suffering for years and years? And there's a switch when the IgM turns to IgG, and something about Borrelia able to something do with this switch, and it's not happening. It's crazy. Of course, these patients say, the doctors, oh, your IgM is still very high. Oh, you got bit again. And I was like, they told me that actually, not because of IgM, but because I didn't have, I couldn't recover fast enough. And I said, no, I couldn't even go outside. I don't think so. I'm bit again. So anyway, so what I had to say is that the immune system connection to Borrelia infection and on the immune suppressive properties of the tick saliva and the Borrelia itself, it is mind blowing. I listened to so many lectures when they looked at the immune system, and I do teach immunology, so I should be able to understand. But it was so many strange things happening in this experiment. My mind was just like, I don't understand what's going on here.
So, Professor, I'm wondering if the biofilm conversation we were having gives us clue into why we're seeing IgM repeatedly, even if you're not getting reinfected. Because what may be happening is when the bugs come out of the biofilm, they're now a new type of bug because they've had this exchange of proteins with all of the other bugs that are in the biofilm, and now your immune system is reacting to it as if it's a new infection because it's seeing a new bug as opposed to the one that it was responding to before.
Yes, I was thinking about this theory, and I think we have to revisit that, because that definitely is this IgM, extreme high IgM titers is just crazy in these patients.
Yeah, I think your biofilm discovery is going to be the gift that keeps on giving, right? I mean, I think the more research that that you do and your colleagues in the research community do on biofilm, I just think you're going to see that really be the gift that keeps on giving. So can we talk about biofilms and whether or not the immune system can break down biofilms? Because we've heard from other doctors that we've interviewed on this program that the immune system generally can break down biofilms. And it's actually when we have immunocompromising events, or the immune system is compromised because it's overwhelmed by all these bugs, that the immune system loses the capacity to break down the biofilm. What are your thoughts on that?
Yeah, I was thinking about that. Obviously we have macrophages, we have those T cells and other lymphocytes. So we have a complement system, obviously. So there is, when you look at immunology, this is mind-blowing what our immune system can do. So I always thinking, why is this biofilm such a challenge for them? Of course, you could say it's multiple layer, like the city, the walls, and everything. But I mean, we have macrophages; they can eat a lot of stuff, and it is something about, I think, what we need to better understand, how maybe whatever comes out from this biofilm either mimic something else or take the immune system to a direction which obviously not gonna attack the biofilm. This is definitely has to be the next big investigation, what's happening here.
So, Professor, are you suggesting that your thought is, although we need to do more research, that the biofilm is sending out these little soldiers to confuse the immune system to not be able to conquer it or mop it up with the macrophages, is what I'm hearing from you?
So we have one paper a couple of years ago. This is when we infect the neuron cells, but we're not just infecting neuron cells. We made these little vesicles. So Borrelia can make this they call the outer membrane vesicles, which practically is the outside the membrane is sort of folding off, putting whatever they're putting there in the bag, okay, and just sending the soldiers, okay. Not even soldiers, because they're just particles. So I never forget it. It was an electron microscopy image, one Borrelia, one spirochete, and electron microscope can visualize these little particles. And it's like, just think about like a wedding gown. It's like it basically went beyond, the whole structure of the spirochete, and looks like it's like this spitting out this thousands of vesicles. So my idea was, why are you doing this? What is the advantage of it? What is in the vesicle? Actually, there's a very, very nice European paper just published last year that they said it has a lot of things in these vesicles. So I think that sort of like, just think about it, sending these vesicles everywhere, it's confusing the immune system totally. Okay, they're going after they're going after the wrong thing. Okay, now we also know these vesicles actually can penetrate cells. Okay, so that was the paper about what happens if you infect these neuron cells with spirochete, the Borrelia, or just sending the vesicles. And what we found, that very similar events happen in neurons. So this called the OMV, outer membrane vesicles. I think that's one of the things. I have a new student who fascinated by that, and she's gonna explore this a little bit further. She wants to see what exactly the function of these vesicles.
That's wild. So that's just a spirochete itself. Never mind once it forms a biofilm, it's even more powerful than just a regular spirochete, right?
And is this vesicles coming out from the biofilm? We don't know. We don't know.
I'm so sorry. My science knowledge is very limited. What is the word you're saying that the varicose? Can you just spell that? 'Cause I'm just fascinated by that term.
Think about just little balls. Okay, it has membrane surrounded. Okay, and now we know that inside they have those OSPA, OSPC. You heard about inside.
Oh, the outer surface proteins. Yes.
Yes.
Yep.
The outer surface proteins, some other unknown proteins. Looks like it's like it is packed to do something. Okay, so we still have to understand what is packed, what exactly the function of. Is it just sort of confuse the immune system, or it is more?
So Matt, remember when Dr. McDonald showed us the slides of the bacteria being like really dirty, like spitting dirt out and spitting all kinds of stuff out?
Yep.
And he kept saying it's like a really dirty bacteria. And I was just wondering, Professor, whether or not maybe one of the functions of this bacteria spitting out so much dirt, and maybe all this dirt coming out of the biofilm is to make you toxic, so that your immune system is not able to function because of the toxicity levels that are developing with all of this junk that's either in the cells or out of our cells, and just sort of overwhelming us with garbage that we're going to need to be able to process either through movement or through saunas or through other detox methods.
It was always controversial whether Borrelia has toxins, like other bacteria, but this little balls, little vesicles. Dr. McDonald told me, and I'm sorry, I forget how does he know it, that these vesicles has a lot of high manganese content. Manganese. I said, okay, so what does it mean? He said, if it's very high manganese, actually it's toxic to the cells.
Mm.
And he said, if you look at manganese toxicity and Lyme symptoms, they are really, really connected.
Wow.
So I don't know that he furthered this. Maybe next time you talk to him, maybe ask him about that.
That's mind-blowing as well, man.
But definitely, as you see, it's like too much going on here. Yes.
Yeah. So they in this battle between the bugs that get spit into us by the tick and the immune system, seems like these bugs have these little bombs that they get to send out, and they're destroying the cells.
Yeah, the brain fog. I don't know who had brain fog.
That's a great transition, because I wanted to ask you about brain fog.
Oh, brain fog. That was bad. That was bad. That was like you behind the curtain. Yeah.
Yep.
Yeah. That's finally cleared up, but it took me a long time. And again, back to this. I don't know that this is coming from this manganese toxicity or any kind of toxicity, because it feels like your brain is toxic. Yes. So I see a lot of people have a lot of speculation what is a brain fog for, but one thing I have me is the detoxification. So it's definitely, that's why I believe that's some kind of toxin, but I don't have proof for it. Okay, just let you know.
Just based on your personal experience,
It's very personal,
anecdotally,
yes.
you believe.
And
you
many
believe
others in the community as well, though, have supported that.
That's what was happening with you. Can we talk a little bit about fatigue now? I mean, you talked about the fatigue that you had and how fatiguing this experience was. And I'm just wondering whether or not the toxicity issue gives us some insight into why it's such a fatiguing or fatigue is such a powerful element of this journey. Do you think toxicity may be the answer to that as well? Or do you think maybe, because our cells are our power plants, right? We have mitochondria that are our power plants.
Yeah.
Are we fatigued because the bugs are competing with us for resources, so we don't have enough resources? Is it because we have biofilm inside of our cells and it's interfering with the ability of the mitochondria to work? Or is it a combination of toxicity, competing for resources and not having resources, and this biofilm inside of our cells just gunking up the capacity of our cells to create the energy that we need?
Of course, fatigue is number one symptoms. Yeah. I think it's multi-level. Again, I think the immune system probably trying to kill this bacteria and working extremely hard. Who knows, the toxic vesicles, make or body very, very dirty. Yes. And now we know that it can actually inside of the cells can be Borrelia. So we don't know how it affects mitochondria or any other very important organelles inside the cells. So I think it's multi-layer. That's why I think the fatigue almost is the last one it leaves. That was my experience. And I talked to people and they said the fatigue is gonna stay for some time. Even other symptoms looks like it's lifting.
Yeah, so I mean, it is, Dr. McDonald always calls us a diabolical bug, and I have to tell you, I mean, the more we're talking about it, I mean, the sophistication of this ancient, bacteria is mind blowing. The way it functions and the way it works, in conjunction with so many other bugs and processes, that it is very, very difficult to treat. It just simply is. Another one of the things that I wanted to put on your radar is I saw recently that I think it was Dr. Zhang's research, where because the Borrelia is a corkscrew bacteria, it actually screws into different parts of your body, right? And then what happens is when the immune system or some other tool cuts off the head of the top of the corkscrewed bacteria, what happens is because it's embedded in other parts of the body, what it does is the head just sort of grows back on again, right? It survives because it's screwed into into tissue or screwed into something, and you're only sort of like cutting off the top of it, but like grass, it just grows back again. And it's one of the reasons why it's persisting. So maybe this is a good transition to your persister conversation, because another place where you're one of the leaders in the world on this topic, because again, we had this general belief in the research community that Lyme could not persist. And it was some of the work that you've done that, where, again, being the black sheep that you are, we started to get clues into to persister, and a lot of people have sort of followed now in your wake, where they're proving that persister cells exist in the monkey models and in the mouse models. But you were one of the first. You were one of the first people to argue for persister cells. So can you talk to us about that and how you came to those conclusions with that research?
So when we treat the bug with different antibiotics, it looks like everything is clear. And I said, oh no, let's just wait. Let's just wait here. So you remove the antibiotics. Sometimes you don't even remove the antibiotics. Just leave the culture on. And 1 week, 2 weeks, they come back beautifully. I have one experiment because at this point I was like, no, we have to kill this bug. There's no way. So it was a culture. It has some aggregates, some biofilm, sort of a mixed culture. I said, okay, we're gonna take this 3 weeks period. We're gonna add doxycycline every single day. Every single day. For 3 weeks. And let's see what happened. So 3 weeks later, we have a stain which stains live and dead cells. I said, I want to see just dead cells. There's no way. There's no way that anything can survive here. And I have this figure, I always show it in conferences. 3 weeks later, yes, a lot of dead cells, it's red, so good, okay. But you could see green spirochetes. You could see some green, some green aggregates. It was mind-blowing. So at this point, I said, okay, you always have to check long-term. So when we did the experiment, we stopped the treatment, whatever it is, stop. I said, now we wait, what happens? Are they coming back? And they always came back. There was a paper, Dr. Eric Hodzig, Emil Hodzig, sorry. He treated infected mice 30 days, and he monitored some molecular bio method, what happens inside the mice. And first it looks like everything is gone. And so, but he's a very patient man. So every month he's checking the mice. By month 8, again, show signs that Borrelia coming back. So that was 2014. When I read the paper, I said, that's it. Okay, we've proven we are persisters. Now it's 9 years later, we still argue on that. Yes, we still argue on that. So I don't know. I mean, of course, Monica Ambers had a lot of interesting study with the monkeys. I don't know how much more we need to tell people that Borrelia can persist.
Well, Professor, there's over 700 peer-reviewed scientific journals and articles proving Lyme persists. And we actually have every single one of them on our website at tickbootcamp.com, because again, it is mind-blowing that people are still debating this. I mean, you've proven it. Other people, you were the first person, you broke the mold, you were the controversial person. 700 other people have proven this, but yet we're still having this debate in the medical community. I don't want to lose sight of your work that you're doing now, right? Because you're now transitioning your original breast cancer research and your Lyme research, and now you're bringing it back with your current research to investigate Lyme and breast cancer. And something that really fascinated me, I just want to read this for our listeners, is you said that you'd heard of people who suffer from Lyme disease and then receive a breast cancer diagnosis soon after. You wondered if the Borrelia bacteria was in the breast cancer tissue. You said your research group examined slides with several kinds of breast cancer as well as healthy tissues. The normal breast tissues were completely negative for Lyme disease, and you found evidence that the Lyme disease was present in the breast cancer tissues, right? So it sounds like you've made a connection here that whether it's a big chunk or just a small subset, there are a portion of breast cancer patients, and you said earlier on, endometriosis and cervical cancer patients who have Lyme disease potentially as the trigger causing that cancer, right? I mean, this could be controversial as well, but you're always the person to bring this controversial stuff to light, and it comes true. So I want to hear what your thoughts are on this.
So think about cancer. So why we die from cancer? If the cancer would stay in place, okay, a good surgeon just cut it out, yes? The problem is spreads. And I was like, your own cells suddenly spreading. Why? What's the reason? I suppose you said, oh, baby, nutrition. Somehow I never really, I saw something else is behind. And actually it is not just me who is now black sheep, but there are a couple of very recent papers when they see that not just Borrelia, other bacteria can go inside the cells, inside of normal breast cells. And making changes, that's what also we see when we infected breast cells with Borrelia, making some changes which can actually trigger metastasis. And I said, why is that? And after it's like, of course, what a bacterium wants? Spread, yes, always, okay? And now you have, I always call the zombies, okay? The zombies, you can go in and sort of rewrite the whole thing, hide inside cells, you save that. Actually, if you know, breast cancer cells also does a very similar thing than Borrelia. It actually revives the environment and make it immunosuppressive, yes? That's why we have now the immune therapies for cancer therapies. And so this is another paper, they showed that they actually making changes that the cells can faster move, finding the blood vessels, get into the blood and spread around the body. But that's exactly what bacteria wants to do. Okay, so when I seen this that Borrelia can able to go into breast cells, and actually we had evidence. Is it lab evidence? So it's not human patient, but it makes it more aggressive. It was our first paper. So
it makes the breast cancer more aggressive. You're saying,
yes,
right?
even a cancer which is actually triple negative. That's a bad one. That cell line was triple negative, already aggressive. We put Borrelia and became more aggressive, which was like, it was
Professor, what does more aggressive mean? Meaning the cell division is faster?
Actually, it was on the cell division, which was very interesting because that's what you think that growing faster. No, it was, we had a technique which measures how fast the cancer cells go through a membrane, because that's the invasion and metastasis. So we used this test, and we infected cells and uninfected cells. And it was like a huge difference. It's in the paper if you want to look at it. And we had some other techniques. Of course, we had some molecular techniques to see those cancer genes are activated, and they are activated. So the most important what we see, it is a protein which cleans, if you want to move, you have to be sure that you have room, yes?
Right.
So there is a protein called metalloproteinase. It's a protein which does that. And the most significant result was that Borrelia able to upregulate this protein so the cancer cells can move.
So it looks a lot like what's happening when the bacteria is in the tick, because what's happening when the bacteria is inside the tick is the protein exchange supercharges the bacteria when it comes into the host, and it becomes a more powerful bacteria. And it looks like when that bacteria gets inside of cancer cells, it has the same impact. And I don't know if the process is the same or not, but the impact is the same. And we now have a more aggressive cancer. Now, of course, we have the other piece of that, of course, which is the immune system is being compromised by the battle that it's having with Lyme, and its shape shifting, and its reformation, and it's working with all these other bugs and spitting so much garbage into us and making us toxic. So it's sort of like the worst of both worlds, where it's supercharging the cancer cells, and then it's having an impact on the capacity of the immune system to manage the cancer cells in the first place.
This is obviously we would love to see what exactly this process. So right now, 2 paper is final revisions. What we did, we infected these cancer cells, and we did something called RNA sequencing. So practically what we did, we sequenced everything what is expressed in the cells. And the results are super interesting. Like chemo-resistant genes are showed up, genes for, of course, inflammatory markers showed up. Right now, too much information, actually. This is our problem. So 2 papers are going out the next month to looking for these different markers to better understand what's happening here. And you mentioned this, Matt, the tissue paper, the cancer tissue paper. We spent the last couple of weeks going to this fancy Yale microscope center. They have amazing microscope there, and to be able to visualize what exactly happening inside the cancer cells, especially those we found positive with Borrelia. So it will be a ton of amazing images gonna come out. Probably, like, we finishing the semester right now at UNH. So probably I'm going to spend the summer to write up this paper. So it will be hopefully published very soon.
Well, that's really exciting. That is really exciting.
Yeah, one of the things, Professor, that I found very interesting that you had quoted, and I wrote this down, is that you believe breast cancer is a combination of genetics and environmental factors. And that's the second time we've heard this in relation to Lyme and other things, right? So in the autoimmune world, we have genetics, and then we have environmental factors. And environmental factors can be the bacterial infection itself, Lyme disease. It can be things like mold toxicity. It can be things like heavy metal toxicity. But when you have genetics that are predisposed to certain conditions, whether it be cancer or autoimmune, and then you have Lyme, it's almost like dogpiling again, where the bacterial infection is supercharging the gene to say, I'm going to express you this way, and you're going to get really sick from cancer, and you're going to get really sick from all these autoimmune diseases, right? And together, it's just sort of dogpiling on your body. So it's so multifaceted. And the way Rich said it was, I think, that 3D chess game that you have to play. And it sounds like I never thought about it. Cancer is the same way, where it's just genetics and environment, where environment can be an infection of Lyme or a wide variety of other tick-borne illnesses, right?
Absolutely, absolutely. I teach genomics, so I will teach, or genome is 99.9% is identical, but those 0.01% what makes us unique. And unfortunately, when I say unique is, too sensitive to certain conditions. Okay, so I know we talked about some people get tick bite and they're fine, and some people just cannot recover from it. So there were some studies showing these different changes, in our genome, and try to correlate it with who gets, chronic infection. But I think that also needs to be a little bit more explored, because a doctor, if you get a tick bite, maybe do a quick panel on you to see if you the lucky one, yes, or you will be the one who might need a little bit more serious treatment and follow up.
Yeah, so I mean, so if there was some genetic testing that could be done at the same time that, or maybe even earlier, then we could do any type of testing for the disease. You may treat that one patient much more aggressively than you would treat another patient.
Absolutely. I mean, this is called a personalized medicine. And I'm teaching genomics right now, I don't know, more than 15 years. And I remember 15 years ago, guys, the personalized medicine is coming. It's still coming, unfortunately, 15 years later.
Yeah.
So I would like to see more, to really taking into your genetic profile when a physician makes decision about your treatment.
Well, you're teasing me right now, and we promised you we wouldn't keep you on for more than 2 hours, but I would love to have you come back so we can have that conversation about genetics and epigenetics and the adaptivity of the, of humans. That I would love to do that, but I'm not going to take you there tonight. As much as that would be an additional birthday present that you could give me. And it's not my birthday yet, so maybe we'll have to have you come back in June.
No, we have to find your birthday. Yeah.
we can have that genetic and epigenetic conversation, because that is something we've been spending a lot of time, Matt and I, thinking about and talking about. So we are within 7 minutes of our commitment to you that we wouldn't keep you more than 2 hours, and this has been fascinating. So we do have to spend another couple of minutes thanking you. And the reason we have to spend another couple of minutes thanking you is because you are literally changing the world. You are putting people in a position where they understand the disease that they have, and we're redefining this disease not as an infection that's caused by one bacteria, but it is a polymicrobial infection. You have changed the world because you're showing why this is a chronic disease and what the persister basis is. And you're putting us in a position where we're getting closer to understanding this disease, so that we can start to come up with solutions, not just individualized solutions, which of course is the dream, but at least some general solutions for people who are in the general population. And we absolutely love you for all the work that you're doing and all the help that you're giving. And people like Matt would not be in the position they were in if it weren't for you. So we can't end this podcast without gushing over how much we value you and the work that you're doing. And if you weren't a black sheep and you weren't a tinkerer, and by the way, if you had money in the beginning, remember, you came out and you had no money, so you had to be creative and resourceful. And because you were creative and resourceful and had to be, you made the discoveries that no one else had. And now you've built the foundation that everybody is building on top of to get us to a point where we can understand this disease and start to treat it. So we just can't thank you enough for everything you've done.
Yeah, I just want to add to that, because on behalf of the entire chronic Lyme community, I mean, look, in several years ago, the estimate suggested we were over 2 million in the United States alone, chronic Lyme patients. And in the world, we know that number is probably larger. So there are millions of us that are looking to improve our health and struggling because there aren't regular doctors who can help us. And it's research like yours, Professor, who are allowing us to get our lives back. The health I've been able to attain because of your work has been miraculous. And again, I wouldn't be where I am if it weren't for you. And many people who have reached remission and have gotten their health back wouldn't be where they are if it weren't for you. So I just want you to know how many lives you've saved. We're talking millions and millions of people that you've either saved or are going to save through your research. So I am just filled with hope and joy. We knew this was going to be a brilliant episode. It was far better than I thought. And I'm getting emotional, but we need more people like you. And thank you so much, Professor.
Thank you for your kind words. And again, as I mentioned, I'm training those students, and I try to, spread this knowledge as fast as I can, not just through publication or presentation or even podcast, but that those trainees who understand chronic diseases. Maybe they're not going to work in Lyme disease. Maybe they work in who knows? Maybe they work in COVID. Chronic COVID now is a big thing. Yes. So, but we have to create this kind of mindset that we don't know everything, and we have to be open-minded.
Thank you for listening to your Tick Bootcamp interview with our guest, Dr. Eva Sapi. To our listeners, we have a call to action. First, if you want to learn more about Professor Eva Sapi, please visit her website page at newhaven.edu. Second, if you enjoyed this episode of Tick Bootcamp podcast, please share it with your friends on social media. Third, Tick Bootcamp is created to Tick by Blueprint. It has been inspired by the information been shared with us by past guests on this podcast. We urge you to visit our website at tickbootcamp.com/bite to view the blueprint. Fourth, don't forget to subscribe to this podcast on Apple Podcasts, Google Podcasts, or Spotify to get your automatic episode updates of our Tick Bootcamp podcast. Please take a minute to leave us an honest review on the podcast platform of your choice. And finally, if you'd like to search our podcast library of almost 350 episodes, subscribe to our email list, or share feedback, please visit our website at tickbootcamp.com. Thank you, as always, for listening.